Γεια σας, καλησπέρα. Λέγομαι Μαριάννα Καψετάκη, είμαι ειδικευόμενη νευροχειρουργική στην Κρήτη και σήμερα θα σας μιλήσω κυρίως για τις μελέτες που έκανα στο διδακτορικό μου και στο μεταδιδακτορικό στην Αγγλία, στο Λονδίνο, στο Imperial College London και στο University College London. Ωραία, τέλεια. Λίγο. Ναι, θα σας μιλήσω για τη μνήμη. Αυτό ήταν το κύριο μέρος που μελέτησα. Και σε υγιή άτομα, αλλά και σε ασθενείς με εγκεφαλικό. Και ο λόγος που εξέτασα ασθενείς με εγκεφαλικό είναι, μπορείς να καταλάβεις έτσι, βλέποντας πού έχουν τις βλάβες, σε ποια περιοχή του εγκεφάλου, μπορείς να καταλάβεις ποιες περιοχές του εγκεφάλου είναι πιο σημαντικές για κάποια είδη μνήμης. Σήμερα θα επικεντρωθούμε κυρίως στην επεισοδιακή μνήμη, άρα τώρα θα το πω λίγο στα αγγλικά. Βασικά επέλεξα να το πω στα αγγλικά επειδή οι πιο πολλές ορολογίες είναι πιο γνωστές στα αγγλικά, ειδικά σε έρευνες και αυτά, σε ερευνητικές εργασίες. So these are the affiliations the institutions I'm affiliated with now. The University of Athens, where I do research, and the Hellenic Open University, and the University of Crete is where I teach neuroscience and neuroanatomy, and Pagani is the hospital where I work as a neurosurgery resident. Okay. Okay. So yes, memory is one of the most important things we have in our life. I believe that, I think. And it shapes many things, and it shapes our personality, and our goals, and our actions. And it's essential for undertaking many everyday activities, like finding a way to our university, or a way back home, or navigating in an environment, remembering how to drive, remembering different words, learning languages. It's involved in many functions. So what are the stages and the different types of memory? Brenda Milner was a psychologist. She's still active, so she's around 100 years old now, almost 100 years old, and she's still active in research. And she provided the first experimental evidence that memory may be divided into multiple systems because she examined Henry Molaison, that's the person next to her. He was a patient who had refractory epileptic seizures, so that meant that they didn't stop with medication. So what they did years ago is that a neurosurgeon would operate on the brain and take some parts of the brain out. So what the surgeon did, you can see in the top picture there, they took out the medial temporal lobes and some other regions, but these were the main parts he took out of Henry Molaison's brain. The surgeon did treat his seizures, but very interesting things that Brenda Milner found during various testing sessions with Henry Molaison was that he could learn new skills, but not new events. So, for example, he could learn. It was like a snail. They were trying to draw a snail by looking at a mirror, and that's quite a difficult task. He could learn that, but he couldn't remember. Like he did it yesterday. He couldn't remember the previous sessions. He did that that drawing session. He could maintain information for limited time, so what we call working memory, but not for a longer time period. And he remembered details of personal events that occurred before his surgery, but not those that occurred after his surgery. So this is sort of a simplistic way in which different stages of memory, how we understand them now. So we have the sensory input. It could be anything like through the eyes, by touching things, or smell, or whatever. Then if we pay attention to that stimulus, then that is kept in working memory. For example, I might tell you to remember 7 numbers, like a phone number. So that's kept in working memory. And if you rehearse that, then it sort of usually goes into long-term memory if you're a normal person, right? I mean, you don't have a disease like, I don't know, Alzheimer's or any other neurological disease or psychiatric disease. So yes, and then from long-term memory, you can take it back and use it for various tasks. And long-term memory, we can separate it into different categories. So the two main categories are non-declarative and declarative. And what we're going to talk about today is episodic memory, which is part of declarative memory. So we have episodic and semantic. So the difference between them is that episodic means remembering events, whereas semantic is remembering just general knowledge. And we can subcategorize these two into episodic autobiographical. So that's got to do with remembering events that have got to do with ourselves. For example, when our first child was born. And then the other category is episodic non-autobiographical. So that's, again, events, but it's not so much to do with ourselves. So that's, for example, when some neuropsychologist tells you to remember words or pictures. It's not really much to do with yourself. But it's, again, an event. Whereas semantic, I said, is general knowledge, and if it's autobiographical, it means general knowledge about yourself. So, for example, my name is Mariana, my address is that. Whereas semantic non-autobiographical is, again, general knowledge, but not relating to this ourselves. So, for example, the capital of Greece is Athens. And the most commonly used definition of episodic memory is by Tulving. So it's memory of personal events. Okay, he doesn't really differentiate here the autobiographical versus the non-autobiographical I said before. But the most important is that it has information about what happened, where it happened, and when it happened, and many details about this particular event. And many studies have shown that the self-perspective is very important in episodic memory. So if you remember things from your own eyes, so for example, this picture down here, usually you remember more details about the event. Whereas if you see yourself in the memory, so like a spectator, this is related to not remembering much information about the event and not being very confident about that event. So how is episodic memory tested? It's usually these two ways, either by recall, so this is without a reference. So I tell you which words did I tell you in that previous list, and you just name whatever you can remember. And the other one is recognition. So here I tell you some options, and you choose from these options. Of course, as you can see, recall is more difficult because you just have to remember it from the start. So how can we test memory? These are very commonly used tests, the general standard neuropsychological tests, which include some memory aspects. I think everybody's familiar with these, the ACE, the mini-mental state examination, and the MOCA. And as you can see here, although we have, down here it says this is the one that tests memory, you can see from all these other pink ones that although people say they test visuospatial and naming, you can see that they do include aspects of memory. So in order to say that's a lion, you have to remember what a lion looks like, right? And in order for the first task, you have to remember how the alphabet goes and the order of numbers. And the same, that's the continuation of the MOCA here. So again, you can see all these the pink ones, although they're not specifically said to be test memory, they do include memory aspects. And so the more accurate ways of testing memory is to have a test which is particularly a memory test, not including other cognitive functions. And episodic memory tasks can vary a lot. It can vary on what you're asked to remember. So is it verbal information like words? Is it spatial information like where things are around you? or temporal order information, like in what order did things happen in whatever they showed you on the screen, for example. And also they differ in how many features do they examine. Do they examine only the words? Do they examine where the word was placed on the screen, or in what order it was placed on the screen? And also the dimensions of the stimuli. So is it just a picture, a two-dimensional picture on a screen, a computer screen? Or is it 3D-like object, or is it an actual object on a table? And also the space in which they present. Is it on a paper, on a screen, on a real table, real environment? And also the interval duration. So between encoding and the recognition or recall phase, how much time has passed? Is it just 10 minutes, an hour? Is it the next day and you're going to be tested? So here's an example of an episodic memory task where it's just words, remembering words. Here's one where you're shown words on the screen and asked to recall them afterwards. Here's one with pictures. So here you're asked to, for example, the first one shows you very small details you have to remember, which exact butterfly it was, in which orientation was the stapler shown. And again, the bottom, in B, again, very small details you're asked to remember. And here's one example from my postdoc project where we looked at 60,000 images. And we found that the more an image looks like a real human face, so the further along there, the more memorable the image is. So we had like real human faces, we had faces that looked like real human faces, like robots or sculptures, and then we had animals which looked like real human faces, like dogs, cats, lions, and then animals that don't look like human faces, like butterflies, bees, like that. And then we have another interesting category, face pareidolia, the one with the car, where it's random objects that somehow look like a real human face. For example, that could be the eyes and the mouth at the bottom. And then just random objects that didn't look at all like a real human face. So that was an interesting study we did. And then we said episodic, if it's related more to the self, we call it autobiographical. So this is usually tested using like questionnaires, asking you if you can tell me about an event that occurred in childhood, for example, and tell me all the details of that event. And these questionnaires can differ a lot depending on how many lifetime periods I ask you about and whether I give you some cues to help you remember, and things like that. And these are the main differences between the episodic memory, which are usually laboratory-based tasks, and the autobiographical memory tasks, which I said is like more like questionnaires. The main difference being like it's okay the dimensions of the stimuli. So the autobiographical memory tasks are usually the encoding phase is a real environment, right? You were in a wedding, you were in your birthday party. Whereas episodic memory laboratory tasks are usually in front of a computer, and you're asked very fast after the encoding phase. You're tested very fast afterwards. So let's go on to my studies that I did during my PhD. First of all, this first study focused on spatial memory, so remembering positions of objects. So the aims of this project was to use the 4-mountain test, which is like a spatial memory task, to examine whether stroke patients have impaired spatial memory, and also compare their memory, the stroke patient's memory, with other patients who are known to have memory problems, like Alzheimer's disease, and also use this task to find out which areas of the brain are most important for remembering spatial information, in particular egocentric and allocentric. I'll tell you in a moment what this is, using various techniques like lesion symptom mapping and lesion network mapping. So this is, let me tell you a bit about egocentric and allocentric. So egocentric means you remember the spatial information from your perspective. So for example, this camera would be to my right, the door would be to my left. Whereas allocentric means thinking of the position of objects in relation to each other, not to myself. And many studies have shown that, especially functional fMRI studies, have shown that egocentric processing seems to be more in the parietal lobe, whereas allocentric more in the medial temporal lobe, for example, in the hippocampus. So this is just a schematic view of how these are presented, and it seems to be the transformation between them seems to be in the retrospinal cortex. So a little test, because I know we've been bored with this talk. So try to remember this. Okay, so which of these images is the same place as the one you saw before? Who thinks it's this one? All right, this one? No, this one? Good, very good. So this is the 4 mountains task. This is the task I gave to stroke patients. Ordinal means there's a different order of the mountains. Spatial means that the distance between the mountains is different, whereas elemental means the shapes of the mountains are different. As you can see, all these are from a different perspective, so this is the main challenge. So what we found, I tested 100 stroke patients, so 50 left and 50 right hemisphere stroke, and they didn't differ between them, so they performed similarly. I should point out that none of these patients had aphasia or hemianopia or neglect at the time of testing, and in fact they were worse than healthy controls. And very importantly, they were as bad as some patients who are known to have memory problems, like Alzheimer's disease patients or amnestic mild cognitive impairment. And also with the lesion symptom mapping technique and lesion network mapping, I found out that the posterior parahippocampal gyrus seems to be most important for remembering spatial information, specifically the right posterior parahippocampal gyrus, and the network of regions shown here. So the next task, examine different, apart from spatial memory, examine some extra things, like the perspective. So it was more tailored to remembering the view from which you have seen an episode. The temporal order, so in what order things have happened, and confidence, so how confident you are about your memory. So yeah, the aims of this project was to develop a new episodic memory task that examines all these features, and also assess which areas in the brain are most important for these features of episodic memory. So what I did, I showed 21 pairs of objects on a table to stroke patients, and these patients had a little camera, so we could video record the scene. Exactly how it occurred. And immediately after I showed them, it was in 3 groups. So immediately after I showed them the 7 pairs of objects, they were asked, like in the animal category, which animals did you see, and where were they placed on the table, and in which order did you see them. And then after 2 hours, we had a recognition test where I showed them on a computer screen 2 videos. One was exactly the same scene they had seen, whereas the other one had some differences. It was either a difference in the position of the object or a change in the angle, so this is a change in the angle, or a change in the order in which they were presented, the temporal order. So I found that damage in the right intraparietal sulcus was associated with poorer memory for remembering the order in which they appeared, whereas damage to the posterior parietal lobe or the medial temporal lobe wasn't associated with impairments in remembering the viewpoint from which they can see in the scene or the position of the objects. And we think that this may have been because we didn't have many patients with these kinds of lesions. So we're hoping that future studies will examine more patients with these kinds of lesions and adapt the task to be more sensitive in these specific conditions. Now, a very interesting case. So out over 400 patients I examined in London, stroke patients. Only one had damage to a very specific area in the brain, the posterior right hippocampus. So this is an MRI scan before the stroke. She just happened to have that because she had a headache, and this was just after her symptoms started, so she had some memory problems. So we did various tasks with her, all these, very quickly after she had the stroke. And here are just a few of the results. So in the ACE, as you can see, okay, she was impaired in memory. As everybody knows, hippocampus is important for memory. Also, fluency in language and visual-spatial were impaired. Fluency in language because she was foreign. Although she had lived many years in London, she was from a different country. Here we can see some impairments in story and code and figure and copy in the R bands. In the word, in remembering words, she wasn't just above, just below the cutoff in the delayed recognition of words. And another interesting task we did with her, because the hippocampus is known, also apart from memory, it's also known to be important for navigation through various fMRI studies. And in Alzheimer's patients, that everybody knows that, okay, they get lost. One of the first symptoms is getting lost. So this task is called the Sea Hero Quest, developed by Hugo Spears at UCL and partners. And what you do is that you navigate this little boat around the various scenes. First, you're shown a map with little flags where you have to reach. And then you're just shown this, and you sort of tap the screen to navigate around. And that's the wayfinding part. And path integration means that you go through just anywhere all around the scene, drive the boat. And once you reach one flag, you have to sort of guess where your initial position was. So that's called path integration. So this patient she was within a normal range compared to control people, like who were controlled for age, education, country, and things like that. And in the four-mounted test, so she was, although she was impaired compared to healthy people, she performed similarly to stroke controls. So I mean stroke controls, we had right hemisphere stroke patients who didn't have damage in the hippocampus. And in the task where I placed the objects on the table, when I asked her straight afterwards what she could remember, she wasn't at all good compared to stroke patients, stroke controls, on remembering the order in which the items appeared. And the most important is this part of the task. So this was after the 2 hours, the recognition phase. So although compared to control stroke patients, she could remember which was the correct video, she wasn't at all confident with her responses. So this is her performance, the red one. So she was very bad at all the different conditions. So remembering in the angle one, the change of perspective, in the position, and the change of temporal order. So in summary, this patient showed us that it seems that the hippocampus, particularly the right posterior hippocampus, seems to be important for how confident we are in our memory, also called meta-memory, which is part of what we call metacognition, so judging our own performance. So yes, I would like to thank my supervisors and my PhD and postdoc, all the people who helped with the projects, the funding I received, of course all of you for your attention and for inviting me to this lovely conference. Thank you very much. Would you like some questions now, or are we going to do at the end?
Χαρακτήρα, εξαιρετικά ενδιαφέρον. Όχι, να ρωτήσω εγώ, πρακτικές εφαρμογές σε σχέση με την εργασία σου.
Πρακτικές εφαρμογές. έχει να κάνει. Κοιτάξτε, αυτό το κανάλι, επειδή εγώ είμαι ειδικευόμενη νευροχειρουργικής και αυτό που θέλω να κάνω στο μέλλον είναι functional neurosurgery. Δηλαδή σημαίνει ότι κάνεις. Μία από τις εφαρμογές είναι deep brain stimulation. Και αν μπορούμε να βρούμε ποιες περιοχές είναι πιο σημαντικές για κάποιες λειτουργίες, όπως η μνήμη, ίσως μπορούμε να κάνουμε deep brain stimulation σε αυτές τις περιοχές, να βοηθήσουμε άτομα που έχουν προβλήματα με τη μνήμη, σαν το Alzheimer, ας πούμε. Ήδη γίνονται κάποιες μελέτες στην Αμερική, αλλά είναι σε ερευνητικό επίπεδο ακόμα. Ναι. Αλλά είναι από αυτό είναι το κυρίως πρακτικό, έτσι, που ελπίζω στο μέλλον να βοηθήσει. Ωραία, ευχαριστώ πολύ.